Both Megaline Pro and Seek Ninja allow you to copy a single annotation, all annotations, or a filtered set of annotations from a source sequence to a target sequence. This demo Megaline Pro project contains two closely related Drosophila chromosome X sequences, a source sequence on the top which contains features, and a target sequence on the bottom that currently lacks features. As we begin, the two sequences have already been aligned. This is mandatory before mapping features using Megaline Pro. To map a single feature from the source to the target sequence, all we need to do is right-click on that feature, and in this case we'll choose Map Feature to Bottom Sequence. We can also map all features or a filtered set of them. This time, instead of right-clicking on the donor feature, we will right-click on the name of the donor sequence, and then we'll select Features. We're given a list of options to choose from. For instance, there's an option to map the features to the active consensus. But for this example, we want to map the features to the bottom sequence. Unlike the situation in which we mapped individual features from the top to the bottom sequence, the fact that we've chosen the entire top sequence as our source has caused a wizard to launch. This wizard begins with a feature screen, where we can choose the features we want to include or exclude from the mapping. In this demo, we will limit feature mapping to CDS features which have a note containing the letters RB. To set this up, we'll choose Features Matching and CDS. If we needed something that wasn't on this list, we could also just type it in here. To restrict mapping to features that have the letters RB in the notes, I'm going to check the filter box and select Note and RB. Now, to make sure we find all instances of RB, I'm going to add an asterisk as a wildcard, both before and after those letters. I'll click Next to move to the Options screen. We're going to leave all of these options at their default settings, but I'll explain what options are available. The top sliders let you specify feature coverage and sequence identity thresholds that are required to map a feature. The lower the number, the less stringent the requirements to call a match. If we were working with an organism that did not use a standard genetic code, we could click on Override Genetic Code and then choose from a list of available codes. We could check the feature report box to add a column to the map features report. This column will contain a Seek Ninja expression that describes the bases in the mapped feature in terms of the source and target sequences. Finally, the unmapped interval report checkbox is used to find areas that may need further annotation. Checking the box causes Megaline Pro to generate a report of intervals without mapped features whose length meets the threshold that you type into the box. In this case, we'll just press Next to move on to the output screen. In the output screen, we need to specify a directory and enter a project name for the output reports and logs that are related to feature mapping. And now we just press OK to begin feature mapping. After the mapping is complete, a hyperlink is provided to the directory that contains our output files. These can be opened in any spreadsheet editor. Here, we're looking at the main report using Microsoft Excel. Next, we'll return to Megaline Pro. Let's start by hovering over one of the features in the target sequence. We can see here that the original note did contain the letters RB, which was one of our search criteria, and a second note has been added showing the sequence that we mapped this feature from, including the percent coverage and percent identity, which were both over our specified thresholds of 80%. However, if we go up to the source sequence, we can see that this feature also has a note with RB in it. To explore why this feature did not get mapped to the target sequence, I'm going to jot down the name of the gene, which is GD17482. Then I'll return to the map features report, which I have open in Excel, and I'm going to search for GD17482. As we can see, it is a CDS feature, but it was not mapped because the percent coverage and the percent identity, which are shown in these columns D and E, did not meet our threshold of 80%. To export our newly annotated target sequence, which has just the features that we have specified, we can select it, then we'll choose File, Export Data, Sequences. To preserve the features, I will select a GenBank file format and save it. When we open the file that we just saved in GenBank format, we can see both the original note and the newly added note showing the sequence that we mapped the feature from. and the percent coverage and percent identity. To learn more about mapping features in Megaline Pro or SeqNinja, or for information about any of our other applications, please visit our website at www.dnastar.com or contact us at support@dnastar.com.